so let's try this out. I'm also going to have subtitles scrolling at the bottom, so hopefully that helps you take notes a little better. I'm going to try to not let it confuse me when I do this, but I might as well try it out since it seems like we may be canceling classes in the future due to coronavirus. Oh, that's not, well, oh, hey, look at that. I got coronavirus right. All right, so let's first start talking about E. coli replication with a really simple model. We covered that before the last exam, but I want to go over it again just really quickly. So they have these circular chromosomes. There's 2 replication forks, and then it just goes in 2 opposite directions around the circle. They begin at the same site, at the Ori site, which we talked about when we talked about binary fission, and then you get 2 circular chromosomes out of that. Oh, here's a better description of that. So they start at the same site, they go in 2 directions, and at the end, you have, in this red and the blue, demonstrate the semi-conservative model, and then you end up with 2 double-stranded circular pieces. But what you don't see here is that the chromosomes are in a double helix, and so before any of this replication happens, and this is not how it works in eukaryotes. The replication process is a little more complicated because we don't just have these circular chromosomes. And those circular chromosomes also happen to be supercoiled. And so what has to happen before DNA replication is that you have to unwind this DNA helix. So let's talk a little bit about how to unwind that. So y'all probably don't remember what this is, but if you think of these old-timey phone cords, I used to have one in my house. They were really super coiled, and you could stretch them out really, really far. But if you stretch them wrong, the coils wouldn't reform. And so what the enzymes that replicate the DNA have to do is they have to very carefully unwind the DNA and replicate inside the helix, and then allow that helix to reform after it's been replicated. And so DNA gyrase, which we'll talk about in a second, is the enzyme that removes the torque from the helix so that it can then be copied. So it unwinds that helix and then gets copied from there. So you see, DNA gyrase is removing the torque, and then those strands get separated before they're replicated. Okay, was there anything else I wanted to say about that? I said that. Okay. So let's talk a little bit about the enzymes that are involved in DNA replication. I said that. Okay. So in the process of DNA replication, there are 3 DNA-dependent DNA polymerases. And so first let's define what DNA dependent is, and that's when you need to have a DNA template which will act to determine which base is added to the next position. So there has to be something DNA present in a strand for the DNA polymerase to be active. It can't just make DNA out of thin air. And then you have the polymerases, which are, like we talked about in class, any polymerase is an enzyme that builds a polymer, and so a DNA polymerase is specifically building a DNA polymer. So some important properties about these enzymes is that they'll only add nucleotides to the 3' end, and they need to have that, like I said before, they need to have an existing template strand to build off of. They can't just grab nucleotides out of the cytosol and just build it. They have to have something to build off of so they know what order to put the nucleotides in. Okay. So this first one we're going to talk about is DNA polymerase III. This is the main replication enzyme, and this is the enzyme which will actually build the copy of the DNA. Even though it's the main replication enzyme, it's called DNA polymerase III because it happened to be the third polymerase that was identified. It's not based in their order of importance. So again, this is the main replication enzyme that will actually build the copy of the DNA. We also have DNA polymerase I, which is going to remove the RNA primer that we use for replication. I'll talk about that in a little bit. And then there's DNA polymerase II, which is not used for DNA replication and will only function in DNA repair. So we're mostly going to be talking about polymerase III and polymerase I when we're talking about replication. So let's talk a little bit about how this DNA polymerase III is going to work. It's, like I said, it's the most important enzyme in replication, and it's going to add the nucleotides to the 3' end of the strand. Oh, no. Okay, how did it do that? There we go. Okay, wait, let's go back here. Yeah, so the DNA polymerase is adding DNA here to the 3' end of the strand. And so that's what's working here, adding these new nucleotides here, and then also working here. The blue regions are the nucleotides that are being added by the DNA polymerase III. It's using the opposite strand as a template to determine which base should be added next. And then what it's adding is the complementary base of the template strand. So like we talked about with transcription and lecture on Monday, it's adding not, so if say this base pair here is an A, what it's adding is a T here. If this base pair that's here is a G, it's adding a C here. So it's making the complement of the base strand, because that basically what it's making is the exact strand that's over here, because that's what used to be attached to it. And the same thing here. What this DNA polymerase is adding is this sequence here. So then you got 2 complete copies of the original strand. Okay. Oh, so before you can replicate the strand, and I'm not going to edit out the pauses I keep taking here because I feel like you might need a little break to take some notes. That way you don't have to stop the video. But again, if I'm going too fast and you feel like you have to stop the video, you can just hit pause on all my blabbering and then continue. So you can't just attach nucleotides to a sequence with nothing attached. And so what is going to be attached first, before the DNA polymerase starts adding base pairs, is this RNA primer. And that RNA primer is attached to the template strand with something called primase. It's an enzyme that attaches the primer. So it's just, dang it, it's just a little 3 base pair piece that's added. DNA primase, not primates. Sorry, the subtitles are kind of crazy. Okay, so this little primer here allows the DNA polymerase to attach then and then start adding nucleotides. So you need to have this primer attached before you can start replicating. And you'll see that primer here and here. So there's one problem with this process though. Everything's going really, really great in this direction, but the other direction what's going to happen is it's going to hit a snag. DNA is really, really long and it can't open on both sides. It makes a little bubble in the middle, just like we talked about with transcription. And so what's going to happen is that this one keeps unzipping here, but what's going to happen is that it can't keep unzipping at the other end, and it's going to hit an end. And so what it has to do is it builds this way, stops, waits until this is open, builds this way, stops, wait until it's open. And that's how you build these tiny fragments. So this strand is called the leading strand, where it's just going to keep on going as the DNA gets unwound and unzipped. And then this one that's going to keep hitting a block as it's closing on the other end, this is going to be the lagging strand. So it builds it in discontinuous pieces. So it attaches, builds, hits a snag, attaches, builds, hits a snag. And that's called the lagging strand. These little tiny pieces that are built on the lagging strand are called Okazaki fragments. Those are named after the scientists that discovered them. And I'm just checking my notes to make sure I said everything. And again, on the lagging strand, you're still using an RNA primer and you're still using DNA polymerase to add these base pairs, but it's just doing it in small fragments on the lagging strand as opposed to just going continuously on the leading strand. Okay. And again, a really important word that I don't have on here, but I want you to write in your notes is that DNA is anti-parallel. So you don't, if DNA were parallel, it would be a 3' end matching up with a 3' end and a 5' end matching up with 5'. But DNA is anti-parallel, which means you have a 3' end on one end and a 3' on the other, and then 5' end on one end and a 5' on the other. And so that, because of this anti-parallel nature, that's why we have to, DNA replication happens discontinuously on the 5' strand and continuously building off of the 3' strand. And if you have questions on this, we can go over it again before the exam. So let's talk about the replication fork where all of this is happening. This is also called the replisome. This is the bubble inside the DNA where DNA is being replicated. And so here you can see the DNA gyrase at the beginning that's unwinding that torque so that the DNA can be opened up. You got this primase attaching. It's adding that little tiny RNA primer. And then there it is. There's DNA polymerase III adding nucleotides. And so on this one, the DNA, it kind of works towards where the DNA gyrase is because the DNA gyrase is unwinding as it's replicating. And so here you've got the leading strand. There's the DNA polymerase just zipping on by, adding them in. And then here's the lagging strand where it has to be done. It's going the opposite direction, and so it has to be adding it in tiny fragments. Okay, said that. Oh, and then what we haven't covered yet is the DNA polymerase I, which you can see right here. The DNA polymerase I is going to bind on to the 3' end of the fragment where the primer is located, and then it'll move the RNA primer and replace it with DNA molecules. So you've got this RNA primer that you've got attached here. You will need to remove that eventually, and so on the lagging fragment, it's pulling that out and replacing this RNA primer with nucleotides as it goes along on the lagging fragment. And so that way you don't end up with a gap on the lagging fragment. Oh, and then at the end here, what you have is the DNA ligase, which is another enzyme involved here, and that's going to join the 3' end of the fragment to the 5' end of the previous fragment so that the strand can actually be completed. And so this is all the extra processing that has to happen on the lagging strand because it's being read in the opposite direction of the leading strand. Okay, and I said that. So just to go through this again, on the leading strand at the origin site, the helicase will enter and it's going to begin to unwind the DNA. The primase is going to come in and make the small RNA primer, which will provide the free 3' end of the sequence. This is all happening on the leading strand. The DNA polymerase III will bind to the 3' end of the primer and will begin to synthesize the new DNA strand moving toward the fork. And then as the bases are opened, the DNA polymerase III will continually add new bases. And there's no, again, on the leading strand, there's no reason to stop or reset until it reaches the other end of the DNA. Oops, didn't realize I switched slides. Oh, yeah, and this is a really great step-by-step walking you through the process of how these base pairs are being added. And then on the lagging strand, what's happening is, as you see on the leading strand, the helicase comes in and opens the DNA to allow the binding of the primase. And then but this time the primase is going to make the primer on the DNA with the 3' end facing away from the replication fork. And then once the primer is in place, the DNA polymerase III is going to bind to the DNA strand and begin to make the fragment moving away from the replication bubble instead of towards it. The DNA polymerase is going to continue to make this new strand until it reaches the 5' end of the previous primer that was attached there. And then this time, they're going to dislodge the DNA polymerase III from the DNA strand and move back closer to the replication fork to begin the next fragment. And then as the replication fork continues to move away from the fragment, DNA polymerase I is going to bind to the fragment at the location of the primer on the DNA strand. And then polymerase I will cut the primer out of the strand and replace it with the DNA sequence, like I said before. And it will only be able to replace the primer. So once the primer is completely replaced, DNA polymerase I is going to become dislodged from the strand. And then there'll still be a gap between the final base of the primer and the end of the previous Okazaki fragment. And that's where the ligase comes in, binds to the gap, and joins the 2 fragments together and completes synthesis of the lagging strand. So by the time the ligase is completed filling in the gap, there will be another complete Okazaki fragment which will already be joined together, and the replication fork continues to move along the DNA. And the process for both the leading and the lagging strand is going to continue until it reaches the termination site on the chromosome, which will be at the end of the chromosome. And so for eukaryotic chromosomes, there's many more than one ORI site. If there weren't, this would take forever. And so therefore, the DNA replication is going to continue pretty much the same, but the 2 replication forks are eventually going to meet, or the replication fork will reach the end of the fragment, which is basically when it gets to the telomeres. So you've already done in your critical thinking assignments, your CTAs, you've already read a little bit about what telomeres are. So when it gets to the telomere, the replication forks, that's when it's reaching the end of the chromosome. And the process of synthesis is going to be the same way that we see in the main part. But the last primer for the last Okazaki fragment will be placed at the very end of the chromosome. And that's what you see right here at the end of this image. But right here, the DNA polymerase isn't going to be able to replace the primer because there's no 3' end which the polymerase will be able to add the new nucleotide. So there's no way to replace this RNA fragment that you have at the end. Because this primer is not going to be as stable as the DNA that's in the strand, eventually this will be degraded, leaving a small single-stranded overhang at the end of the chromosome. And because the leading strand will be able to be synthesized to the end of the chromosome. So because this single-stranded region of the DNA is very unstable, this will eventually fall off, and the final new chromosome will be very slightly shorter than it was in the parent cells. As you keep replicating over generation to generation, as this happens too many times, the DNA is going to actually start removing genes from the chromosome. And if the chromosome becomes too short, then the cell will recognize that shortening and send that cell off to greener pastures to die. But you've already read about how you can fix that shortened telomere with the action of telomerase. So what the telomerase does is it's an enzyme complex with an internal RNA primer already bonded to the protein. And so it's already got that primer in there, so you don't have this piece that can't get fixed. And then that primer is going to contain a short repeated sequence, which will be able to bind to the repeated sequence on the leading strand of the DNA. And then by increasing the length of the repeats at the end of the chromosome, the DNA won't become shorter with each division, and therefore you're not going to need to remove the cell from the cycle because the telomerase is fixing that and not making it shorter. And as you read in your CTA, the addition of the telomerase activity may be able to increase the lifespan of the cell indefinitely, depending on the organism, and allow the cell to continue to divide without the loss of DNA at the ends of the chromosome. Because this continual lengthening of the telomere will allow the cell to divide indefinitely, the cell could become immortal, which is commonly found in cancer cells. And so if you've ever wondered how they do cancer cell research in the lab, they're using cell lines that are immortal because of this telomerase. And so you can freeze those cells in liquid nitrogen and then bring them out of liquid nitrogen, and they'll just continuously regenerate in the lab, which is really, really cool. Yeah, so some cancers aren't immortal, some are. There's any cancer cells, so like the Henrietta Lacks cells that we can talk about later in class, those are an immortal cell line, perhaps because of the action of the telomerase, but not all cell lines are immortal because they don't have this action of the telomerase. And that's it. Okay, so that covers DNA replication. I hope that I covered it in enough depth for you to understand how it works. It's really, really important to understand how this works because DNA is the stuff of life like I've talked about in class. So please watch this video. Take notes while you're watching it. Take notes on everything that I'm saying because I'm sure these subtitles have not been getting every single word that I've been saying correctly. So make sure to take lots of notes. And then if you miss anything, you can skip, you can go back. And I really recommend you watch this video more than once just so you get everything down. And then also, as you're taking notes, write down some questions that you can actually ask me in person. Okay, that's it. The end. Finito. I'll see you on Friday.